Good morning, everybody. Thanks so much for being with us today. We have a really great presentation, one that's been being requested for quite some time. Before that, we have our usual weekly updates, and we have a couple updates this week. We'll have more updates coming the following weeks as well, including more on vaccination and so on. But we do have Dr. Ben Pitsky, as usual, with us to give us updates on the Delta variants as there's more information coming out. Dr. Pitsky, thanks a lot for being here, as always, and thanks for providing weekly updates.
Thanks, Errol. Let me put that on full screen. Can you all see that?
Yes.
Yes, great. So briefly, a quick update on tracking the COVID-2 variants, in particular the Delta variant, which we're starting to hear a lot about. So a reminder, Delta is the B.1.617.2 lineage. It now has two additional sublineages that have been termed in the press Delta Plus. They're now all considered one. The main spike mutation in this lineage is L452 to R, which was also found in Epsilon, which is previously known as the California variant. And then this additional sublineages contain another mutation, K417 to N, which is found in the beta variant, also known as South Africa or B1351. From the CDC, their data is a little bit out of date compared to what we are able to obtain here. The B.1.617.2 or Delta variant accounts for approximately 30% of cases across the United States. Sorry about that. And about 35% in California. You can see that it's this orange color. It varies quite substantially across the United States. More recent data reported today from the CDC, but not yet published on their website, suggests that the Delta variant makes up about 50% of cases in the US. Here's our data based on mutation-specific PCR followed up with whole genome sequencing. You can see that the number of genotypable samples has decreased over time, as I've reported over the last several weeks. This is when Delta first came into our population on around the first week of May. At that time, there was still some of the Epsilon variant. However, now all of the variants we see with L452R are the Delta variant. And you can see now that this variant makes up close to 80% of our genotype samples. So the Delta has taken over in our patient population. So with that, I'll stop, and I'm happy to take any questions in the Q&A in the chat. Thank you.
As always, Dr. Pitsky, thank you as well for these weekly updates. We really appreciate it. And then just one other update today, we have Dr. Steph Harmon.
Thanks, Errol. Good morning. A few announcements for this week. I just wanted to call everyone's attention to a great event happening tomorrow, sponsored by the Stanford Center for Innovation and Global Health and the Stanford Center for African Studies. It's going to be a conversation with Dr. Ayoade Alakija, who is the co-chair of the Africa Union Africa Vaccine Delivery Alliance for COVID-19. And they will be talking about, along with Dr. Barry, our Dean for Global Health, and Paul Costello, speaking about vaccine equity and gender equality in Africa. And so that'll be tomorrow, July 8th at 10 a.m. our time. And we'll have the registration link put in the chat. The next event that I wanted to share with you all is the virtual conference, the Women in Medicine Summit. This year is the third annual one. And it will be September 24th through the 25th. And it actually features two of our own members of the Department of Medicine, Dr. Argavan Sales and Dr. Lucy Kalanithi. We'll also put that registration link in the chat. Okay, thanks so much.
Thanks so much, Dr. Harmon. I appreciate it. And then now moving on, before we do, next week we have Dr. Chuchu Desai talking about the topic All Hands on Deck, repair of acute lung injury, followed by discussion on cytokine storm syndrome. We have a presenter who will be joining us from the University of Alabama. And then the week after that, we have Drs. Shulman and Chandran talking about racial disparities in health and healthcare. So I'm going to turn it over now, and I thank Dr. Parsnet, who's joining us from other side of the country. As you can, it might look like her Zoom background in the back there, but it's a real background. She's in a really nice area, and she was able to join us to introduce Dr. Relman. So Dr. Parsnet, I'll turn over to you, and thanks for being with us to introduce Dr. Relman.
Sure, it's a pleasure. So it's a great pleasure today to introduce David Relman. When Errol asked me to introduce David yesterday, it occurred to me that I have known him for longer than most of the audience here today has probably been alive. So I've known David since I was actually in college, and he was at MIT, and I was up the road from him and a friend with his brother. And David did go to MIT and subsequently went to Harvard Medical School. And then we joined up again at Mass General, where we were both residents. So I've known David for quite a long time, and I'll do my best not to go on and on about his many accomplishments, and even more careful not to say anything that would be embarrassing to him or more likely to me. So David, when he was a resident and a fellow at Mass General, a clinical fellow, was one of those residents and fellows that you really wanted to have to work with, because he was always calm and collected and thoughtful and never got ruffled, and could even make you laugh, even in one of the most stressful cases that you were going over. So I always appreciated working with him throughout in clinical medicine, and I know our fellows today still enjoy that opportunity to work with him. But after he finished his clinical fellowship, David came to Stanford, where he worked with Stan Falco and Lucy Tompkins in doing a postdoc in microbiology and the interlink with microbiology and infectious diseases. And during that time, they developed this method of looking at 16S RNA to identify microbial pathogens. And their first work on this was looking at microbes found in what were thought to be sterile sites. And using that technique, David and identified two new pathogens that we're all so familiar with today: Bartonella henselae, the cause of cat scratch disease, and then Trophorema whippleae, the cause of Whipple's disease. And this was a huge splash because people had been looking for these organisms for so long, and he and Lucy and Stan were the first to really identify those organisms. Subsequently, David used that same technique of looking at 16S RNA to look at the microbiome. So really, David is the real father of the field of microbiome research. And he first started looking at the gut microbiome, but then expanded his work to look at the genital microbiome, and then looking at a lot of mammalian systems and microbial ecosystems. And it's just a fantastic body of work. And what makes, I think, David unique in this field is his careful work that he does, and how he doesn't overstate it, and how considerate he is about interpreting the data. And as somebody who has dabbled in microbiome research, I think he is really unique in that respect, at the thoughtfulness at which he approaches thinking about microbial communities, and the thoughtfulness with which he approaches science more generally. And you see that in also his mentees, who have all gone on to these great academic positions. When you look through David's CV, though, what's most striking is all these national committees that he's been on. And again, that's because of just his very thoughtful, careful approach to science, and how great he is at looking at things from different directions. And among the committees that he's been on, many committees he's been on, there have been quite a number that have been on biodefense and bioterrorism. And he's been on committees for those at the NIH, the National Academy, Department of Defense, especially DARPA. He's also been a leader of the ID Society, where he was president. And at Stanford, in those regions of biodefense and bioterrorism, he's also been the co-director of the Center for International Security and Cooperation, CSAC, which is over at Freeman Spogli. David was on the committee that really looked over the anthrax outbreak in 2001, the bioterrorism of anthrax in 2001. And he's also currently leading the committee that's looking on the mysterious diseases that are affecting members of our embassies around the world. Most recently, David has been a vocal, a strong voice for trying to understand in the details and really understand the origins of COVID, and was a senior author of a recent science letter that was encouraging this investigation so that we can understand how to prevent these sorts of events from happening again. And that leads me into his talk today, which I think is going to be fantastic, which is COVID-19 and SARS-CoV-2. So thanks, David.
Thanks, Lucy. Thanks, Julie. Your introduction was very, very generous. I have to say it's been a real pleasure and honor to know you and all of my colleagues here at Stanford for these many years. I plan to talk about a topic that has received a fair bit of attention recently. It's somewhat contentious. There's a lot to say. There's not a lot of time in which to say it. And I'd like to save a little bit of time for questions. So I'm going to touch very briefly on the nature of emergence of infectious disease, some of the early events that occurred in the history of this pandemic, and then put forth a couple of hypotheses about the origin of the pandemic and the virus, the evidence for and against, and then the paths forward. It's quite clear, and I'm sure clear to all of you, that our history as humans, is replete with both images, but also evidence of the profound impact of biological agents and biological threats in causing a great deal of suffering and harm. And much of this was not fully recognized as of microbial origin, but clearly it is now understood to be so. These events that we now recognize seem to be occurring with increasing frequency and with increasing diversity and variety. And here's some examples on this map. Some of this, of course, is due to a surveillance artifact. We're simply better at detecting emerging events and microbes than we were previously. But there are a number of factors that we do believe account for an increasing frequency of these events. And these factors result from a great deal of work that's now ensued in the last 20 years, some of it led by the National Academy of Medicine through a group called the Forum on Microbial Threats, which I had the pleasure and honor of chairing for about 10 years. And the factors are often binned into these three categories: microbe, host, and environment. And I don't want to belabor the details here, but I will point out that within the category of environment, climate and changing land use are factors that are increasingly seen as important today. And in particular, the changing use of land by humans with the resulting increased numbers of pathogen-associated hosts, including bats. Now, the other factor that clearly comes into play, especially in recent years, is human activity. And some of that is viewed as the source of potential threat. So this is, of course, a large area that we don't have a lot of time to discuss here today, but it arises from the incredibly powerful, increasingly prevalent capabilities of humans to work with and manipulate biology and biological systems, almost entirely for the good, but occasionally for the bad. And that then fills in this set of factors with a couple of additional factors that you might want to consider, including the activity of humans in increasing diversity of microbes through synthetic approaches, as well as engineering approaches, and the same thing for hosts, for example, plants, and our creation of monogenic crops that become increasingly vulnerable to pathogens, as an example. So just a brief word about the early events as we know them with respect to this pandemic. The first notice to much of the world came from the Wuhan Municipal Health Committee by way of a ProMed mail. And this was the announcement at the end of December in 2019. These cases had the features of SARS, but also of many other illnesses of a nonspecific sort, suggesting an acute respiratory distress syndrome. The first publications of cases pointed to origins, or at least the clinical events, as originating sometime around the beginning of December, although that wasn't so clear, and in fact still is not so clear. These cases also were sometimes associated with exposure to a seafood market in the city of Wuhan. But as you can see from this plot in this Lancet paper, many of the first cases did not have a known exposure to that market. And questions have arisen since then about the exact timing. This is a paper published in the last year by investigators from the CDC that looked at stored blood samples from blood donations in the United States, looking for evidence of exposure to SARS-CoV-2 through the use of antibody detection. And this paper suggests that a meaningful, discrete number of donors back in mid-December in the United States had evidence of SARS-CoV-2 antibodies, suggesting that their initial infection would have had to have taken place in early December at the latest, and suggesting origins that date back prior to December. And then we have the first description of the virus, which was announced in January, published online in February of last year. And this was a paper out of the Wuhan Institute of Virology, where a great deal of work has taken place on coronaviruses and bat-associated viruses. This was a figure from the paper that revealed the genome of this new novel coronavirus. You can see from this phylogenetic tree on the right that the virus found in a number of patients was a beta coronavirus, and most closely related to a whole variety of previously described, in recent times, bat-associated SARS-like coronaviruses. The nearest known neighbor was a virus that was announced in this same paper called RATG13. And in this paper, they point out that it being the closest relative at 96% sequence similarity, this was a virus that had become known to them several years earlier when they were sequencing parts of genomes from lots of bat samples. And when they say in this paper, when they discovered SARS coronavirus 2, they looked back and found a bit of sequence that seemed to match it, or was close. And they then, they say, pulled out the sample and sequenced the rest of that genome, RATG13, to reveal, again, the now closest known, and still closest known relative of SARS-CoV-2. Now, I'll just say, 96.2% may sound like a close match, but in fact, in terms of evolutionary time, and as best one can date the time of the last common ancestor of RATG13 and SARS-CoV-2, we're talking about tens of years, if not a bit more. So it's still not terribly close. So the questions we have before us are, where, how, and when did the first encounter take place between SARS-CoV-2 and humans? And I'll just point out that there are two closely related questions here. One is about the origins of the virus, and the other is about the origins of human infection with it. So what are the various hypotheses that might explain this first encounter and the evidence for these hypotheses? I would say we can put them into two bins, although there is clearly a spectrum and a continuum across this landscape of possible explanation. The first would focus upon the likelihood of a natural so-called spillover, where the virus moved from a natural, presumably animal host, and because of the phylogenetic tree and the closest known relatives, we would say a likely bat host, and then spilled over into humans. This natural spillover could have taken place at the habitat site of this natural host, say the bat host, for example, in a cave. Or it could have taken place closer to the place of human habitation, for example, in a city, perhaps because the natural host was moved or a sample of it was moved into a place where humans spend more of their time. Now, the second category of hypotheses involve a laboratory. Why? Because we know that laboratory researchers are interested in these various viruses, and in particular have been intensively studying bat-associated viruses by going to their natural habitats, gathering samples, and returning to a laboratory. One can imagine that a researcher became infected, perhaps, during one of these field investigations, and perhaps didn't even realize that they were infected. Another possibility within this second category is that samples, or perhaps the hosts themselves, the bats, were brought back to a laboratory where perhaps efforts to propagate a virus or viruses were productive, but yet unrecognized as such. And I mention this because a number of virus propagation efforts often result in the failure to see any evidence of growth. And in some of those cases, we know that viruses may have grown to a limited degree. We now know that with SARS-CoV-2, limited growth of SARS-CoV-2 in a laboratory that wasn't prepared to handle it could easily have resulted in an accidental infection. And then finally, we have known work that involves the manipulation of viral genomes in a laboratory, and in particular, those of the bat-associated coronaviruses. Okay, the first thing that you might wonder is, what about just looking at the genome of this virus? Doesn't that tell us where it could have come from? And I will say there are two things that people have sought to understand from the sequence. The first is the time since it last diverged from its closest known relative. And I mentioned that RATG13 is the closest known relative, and the time of likely divergence is really decades ago. But nonetheless, one can use various evolutionary methods to estimate when the true last divergence and common ancestor did take place. And this is a paper by Mike Worraby and others that did some really interesting work suggesting that when you look back through clock inference methods, as they're called, as well as look forward with simulations addressing the possibility that the very first case is likely to have not led to sustained transmission, but rather a series of aborted transmission events. That when you do all of that, and they did, you end up with a time of first cases sometime between the middle of October and the middle of November. So this is the kind of thing that you can do with the viral sequence. Another thing that people have attempted to do with it is to make conclusions about whether it shows evidence of manipulation. And this is a paper I want to draw to your attention because it was published last year in March by 5 very eminent virologists and became the most highly cited paper on the origin of the pandemic. And I want to call attention to a couple of statements that they make. First, they say, if genetic manipulation had been performed, one of several systems available would have been used. However, the data irrefutably show that this virus is not derived from any previously used system or backbone that we imagine they would have used. However, this is my commentary, this assumes that all available virus backbones are known to us and are public. The authors go on to say a hypothetical generation of the virus by cell culture or animal passage would have required prior isolation of a progenitor with very high genetic similarity, which has not been described. That's true, but it also assumes that all prior isolates are public. It also ignores the fact that when you grow viruses from a sample that has multiple viruses, and coronaviruses in particular, you very often get recombination that's unrecognized within the growth vessel. This also assumes, their statement, that viral growth was recognized, and as I've suggested, it may not have been. And then finally, they say, since we observe all notable features of this virus in related viruses in nature, we do not believe that any type of laboratory scenario is plausible. Here, I'm still scratching my head about the logic of the statement. It again ignores the possibility of field work where the actual virus was brought back to the lab unknowingly and grown perhaps in an unrecognized fashion, leading to accidental infection. So I don't understand personally the impact that this paper seems to have had in guiding a lot of public thinking, including amongst my colleagues and your colleagues, into assuming that a laboratory could not have been involved in any way, shape, or fashion. Basically, the immediate ancestors are missing, and they may be in the natural environment and they may be in a lab. We simply do not know. The sequence itself does not reveal the virus's origins. Okay, so what kinds of evidence do we have? In the first category, we have 4 bits of evidence. 2 of them I've shown here in summary form. The first is we have history. We have that map of the world that I showed you with all of these numerous events, the vast majority of which are of natural origin. They're spillovers. History is a powerful precedence here in suggesting that likelihoods would argue for another natural spillover. The second bit of, again, circumstantial evidence is that there still is a great deal of undiscovered natural diversity among the coronaviruses. Here's a paper that appeared this past year, published by the Wuhan group, showing the kinds of diversity that they find when they start to delve into this incredible inventory of bat samples that sit at the Wuhan Institute. When they looked across China at the various sites where bats were captured and samples taken, they found the vast majority of this coronavirus novelty was down here in the south. This is Yunnan province here, where the greatest amount of beta coronavirus diversity was discovered. But there's still a great deal more out there, inevitably. The third bit of circumstantial evidence is that we know that there's a great deal of movement of trade, both legal and illegal, of wildlife, of the very hosts, natural hosts, that we think could be the natural hosts of SARS-CoV-2. So these would be, in essence, facilitated encounters. And here's an example of that, a paper that came out just several weeks ago, documenting some illegal wildlife trade into Wuhan with a whole variety of different animals, none of which have been shown yet to be natural hosts for SARS-CoV-2, but of course, some of which could be. And I'll note, though, that none of these animals that were found in this survey were bats or pangolins, another prominently touted possible natural host. And then finally, spillovers do happen more often than we know. And there are plenty of studies that point to this. There's evidence, for example, of henipaviruses. This is Nipah and Hendra virus, and spillovers into human populations in Africa that are revealed only when you take blood samples from apparently healthy individuals in places where you might suspect there's encounter with bats, and find, in fact, that asymptomatic humans do have antibodies against these otherwise, dangerous viruses. So one can easily imagine that there are plenty more ongoing spillover events that are perhaps aborted or unsuccessful, but increasing the likelihoods that a natural spillover could in fact be the cause of this pandemic. And here's just one more paper to make this point. The people at Wuhan did go down to the southern parts of China to look for evidence of serologic antibody against SARS-like viruses. And they looked in particular near a cave where they had been studying a lot of bat coronavirus diversity, again, down here in Southwest China. And from these 4 villages, they found 6 positives for exposure to bat-like or bat-associated SARS-like coronaviruses. The controls here were individuals from Wuhan, where they suspected there was very little bat exposure. And you can see, in fact, there were no seropositives. So simply keep that in mind. Okay, what about the evidence for a laboratory-associated encounter? The first bit of circumstantial evidence is geography. By that, what do I mean? Well, the place where, again, all of the survey work has shown that the greatest amount of coronavirus diversity exists in China is down in the Southwest, as I've mentioned. And yet, the disease first appears, it seems, in Hubei province, and Wuhan in particular, which is about 1,000 miles away. Meanwhile, in the center of Wuhan are the various facilities of the Wuhan Institute of Virology and the Wuhan CDC, which actually is just a couple blocks from the seafood market that was initially identified as a possible source. And these are the very labs where a great deal of bat coronavirus work has taken place, which brings up the second point, again, circumstantial. The known work in these labs in Wuhan. These labs have been a prominent player in what's called the Global Virome Project. This is a worldwide effort to understand viral diversity and in particular, anticipate future viral threats to humans. And the WHO report that came out several months ago about the COVID origins highlights the fact that at the Wuhan Institute of Virology, there are over 19,000 bat samples, 13% of which is positive for coronaviruses. And in particular, amongst the kinds of work that the Wuhan Institute of Virology has undertaken is some work that some would view as potentially risky. And here's an example. This is a paper that was published in 2017. It was work partially funded by the US National Institutes of Health. And in this work, they sought to understand some of these novel bat-associated coronaviruses. The way they did this was to take the key part of the genome of these viruses, the spike protein gene, and to insert it into another bat-associated SARS-like coronavirus, a virus that they had previously studied, so they understood a bit about, called WIV1. So these are chimeric viruses. They're mostly WIV1, but they have a little bit of a brand new bat-associated virus in them. The reason why this work could be viewed as risky is that WIV1, the backbone, their starting virus, was a virus that many in this field had already determined to be a particularly risky virus. Ralph Barrick, a prominent investigator in this field, had characterized it as poised for human emergence. This is a virus that is already capable, coming straight out of a bat, of growing in human primary respiratory epithelial cells. And so these investigators were essentially creating chimeras with unknown potential properties, starting with a virus that many viewed as dangerous to begin with. Okay, third point. Accidents, just like natural spillovers, accidents happen much more often than we know. And I'll point you to an incredible investigative piece by Allison Young that was published over a series of years, several years ago now, in USA Today, where she used FOIA requests and collected evidence of more than 1,500 laboratory accidents in the United States involving dangerous infectious agents, so-called select agent pathogens. In more than 800, workers received medical treatment. And in particular, laboratory accidents involving SARS, the original SARS, were well recognized and consequential. So there were 3 accidents in 2004, one of which resulted in the death of the mother of a laboratory investigator when they inadvertently carried the virus out in an accidental infection and then spread it locally. Finally, unfortunately, there has been a lack of transparency and a number of inconsistencies in the reporting of the work that's been done at some of these laboratories in Wuhan. Now, I don't mean to suggest any motive at all, but simply point to some facts. First of all, we now know that the Wuhan Institute was involved in the investigation of 6 patients that were sick in 2012. We know about them from a master's thesis that was published by a student in China who later worked at the Chinese CDC. These 6 patients had a SARS-like illness, 3 of them died, and all of them had spent a number of days in the very cave where it turns out the Wuhan Institute had decided to go and investigate, and then learned of an immense amount of coronavirus diversity, including the very cave where that closest known relatives of SARS-CoV-2 came from, RATG13. That was from this very cave where these 6 people became deathly ill, 3 of whom died. Now, the reason why this is of note is that they never told anybody about these cases, nor about their investigation of them. And yet their mission was to alert the world of a possible next SARS. Only this past year, when many questions were raised about these cases and about the origin of RATG13, did the folks at the Wuhan Institute issue an addendum to their initial announcement paper of the virus of SARS-CoV-2. And in this addendum, they say, oh yes, in fact, you're right. We did know about some cases. We did investigate them. We didn't find anything. And these were the cases that prompted us to look in the cave that revealed RATG13. But it does raise these questions. And again, much of the data, or some of the data, I should say, that has come from early investigations of related viruses is incomplete and doesn't entirely hold together. For example, the RATG13 short read archive that was deposited does not support the reconstruction of the entire virus genome. And the same is true for some of the other data. Okay, so where do we stand? Basically, all we have is circumstantial evidence. SARS-CoV-2 has not been found anywhere other than in infected humans, and then the animals that humans have secondarily infected, like the minks in the Netherlands, and contaminated fomites, etc. The immediate ancestors still remain missing, and without them, we cannot describe the recent history of this virus. And without them, it's very hard to understand exactly where that first encounter took place. Neither hypothesis that I've described can be ruled in, neither can be ruled out. Many people have opinions about which is more likely. I am not going to share any opinion about this because I think it's in some ways unwise and perhaps unhelpful. The issue has become deeply politicized and, unfortunately, in my view, subject to groupthink. And that goes for scientists as well as other members of the general public. So, what would the elements of a credible investigation look like? There are 2 kinds of elements that you could talk about. One is the data. And this is not meant to be an exhaustive list, but I just want to highlight the kinds of things that we really still need to look at. We need to look at a more extensive archive set of animal samples and their derivative data from natural habitats of suspect hosts, the trafficking patterns and routes, the farms and the markets where trafficked animals end up or originate, the laboratories into which samples are found. The same thing goes for clinical samples and their derivative data, as well as other forms of clinical data, wastewater samples, for example, from Wuhan and other parts of China, social media data, and then the laboratory records, the field activities. Please remember that when we talk about a laboratory-associated accident, this includes any activity by a laboratory-associated person, including their field work, the efforts to rid the lab of unneeded materials, the disposal patterns, the safety records, the nature of the experiments, the inventories of samples and viruses. I'll mention that the Wuhan Institute of Virology played an enormously valuable role in contributing to our understanding of coronaviruses. They used to have a virus sequence database that they shared with the world. It was an incredibly helpful thing that they were doing. This virus database was taken offline at the end of 2019. And as it turns out, it went offline in September of 2019. And for reasons that have still not been fully provided, that database is still not available to us. And for all of these data, forms of data, we will need evidence of veracity and provenance. The other kind of element is the process by which an investigation would be undertaken. And I think this is equally important. We need something that's going to be objective, that's not going to start with a particular set of a priori assumptions, the way that the WHO investigation did. They had a number of a priori prior assumptions and judgments. We need it to be science-based. We need the expertise to be broad and international, and not just inclusive of scientists. We need to represent all of the global constituencies, which now have paid so dearly in the form of consequences from this outbreak. They all have something to say and should be represented. The process needs to be transparent, needs to be independent, it needs to be subject to independent oversight, and needs to be responsibly managed as well to minimize conflicts which have clearly arisen so far. I could have started with this slide, but I will end with it. Why does this matter? It matters because I view it, I think we all view it, as incredibly important to be able to anticipate better and prevent, if possible, the next pandemic. This kind of investigation that I've been talking about may lead to a more effective response to this one. It certainly may limit further recriminations and perhaps the likelihood of conflict. It will advance discussions about risky science. If we learn nothing more, what I fear is that a very problematic and difficult topic, that of risky science, will be again ignored as it has been in the past. A proper investigation now will teach us how better to undertake investigations of this sort in general. And then finally, it will lead to a greater understanding of our precarious, if you will, coexistence with the microbial world. So let me just summarize. We do not know still how, when, or where this pandemic started. Anybody who asserts they know should be asked for the data on which they base their assertion. And while a definitive answer may prove elusive, it may not be easily found, a proper investigation is important nonetheless. Greater clarity, I think, is necessary and feasible. So my thoughts have been published in 2 places at least. I'm happy to talk with any of you about your points of view and any questions. I think this is an incredibly important topic that we not feel afraid to address in a sort of a dispassionate, clear, and objective manner. And so I hope that's where this leads. Thank you.
Dr. Relman, thank you for that excellent, really digging into the information that we know and sharing your thoughts and just really being with us today on this. Obviously, it's a very difficult conversation. It can be controversial as well. Thank you for focusing on the science and the facts at hand and for sharing your expertise with us. So I'll ask people any questions, feel free to ask them. There's a couple that have already popped up here, and I'll just start getting into those. I have one, Dr. Siegel asks, please discuss the role of recombination of which coronaviruses are uniquely capable in generating natural variants.
Right. So viruses in general have a tendency towards recombination, but some families are famous for doing so much more often than others. And as it should turn out to be the case, coronaviruses are one of those families where recombination is clearly common in nature. And in fact, takes place even when coronaviruses are grown in the laboratory or grow in a natural growth vessel like a host, a human being. Recombination has clearly taken place within bats, and bats can be infected by multiple coronaviruses at one time. And recombination does seem to take place in humans as well. So I guess, you know, again, this is a common feature of the biology of these viruses. When we look at the SARS coronavirus genome, it does appear to be clear that at some point in its history, recombination may have taken place between more distant ancestors. But whether we can see evidence of more recent recombination depends entirely upon the availability of those more recent starting viruses, which again, we don't have. So a lot of people have gazed at this genome and said, can we tell whether recombination took place very recently, perhaps in a laboratory? And I would say we cannot tell that.
Dr. Relman, Dr. Owens asks, do you think an investigation such as you outlined will be conducted?
I do think, and I know that a number of investigations are likely to ensue. There are plans afoot from multiple quarters to carry on the work that has been proposed and was attempted by the WHO. The WHO itself has been talking about a phase 2. It remains unclear as yet whether they will, and if so, in what form, begin such a phase 2. But meanwhile, there are a number of governments and within the United States, governmental agencies and private groups that have begun to talk about what an investigation might look like and whether and how, in fact, it should begin. And I'll just point to one. There's a group being led by a guy named Phil Zelikow. There was just an editorial in the Washington Post today about his effort. He has created a COVID planning group, of which I've been a member, but Phil is the person who put together the 9/11 Commission for Congress, and he's been proposing that Congress and or other elements of the US government, and perhaps other governments, get together and undertake a more expansive look at not only how things began, but how things unfolded. And that's just one example. There are, I think, going to be a number proposed.
Dr. Relman, Dr. Weinberg asks, he says, well, thank you for so much for a thoughtful, informative presentation. Do you think that microbial gain of function research should be internationally regulated? Given current state of nationalism, security states, do you think such regulation is possible? In contrast to tools available for IAEA, like monitoring of nuclear activity, seismometers, rogue isotope detection satellites, is it possible to regulate lab work without international cooperation?
Yeah, those are all really, really good questions. I'll sort of premise my comments around what I will call risky research. Gain of function is a term that has been thrown about and used in multiple ways. It's a bit fraught with some confusion. But I think it's a very legitimate question to ask about work that today might fall into this category of so-called dual use research of concern, but really risky research. And I am a person that does believe that this is now a time when we have to acknowledge or certainly examine what it is that scientists may be capable of doing that creates unusual risks. Let me say at the same time that I view the vast majority of science as both immensely important, but also lesser likely to lead to risk, true, genuine risk of a catastrophic type. But now for the first time, perhaps in the last 10 years, it's clear that some research, a small amount, does have that potential to lead to grave consequence. And I worry that we haven't yet clearly defined what does that research look like, how can we define it better, and what should we do about it? How can we mitigate the risks? Part of the problem is we don't have a good governance scheme. We've been asking groups like the NIH to oversee such work when in fact, they really weren't created to do so, nor does their culture support this kind of work. So I think we're still struggling both with a definition of what it is, as well as how we're going to address it. And you raised this interesting question about the IAEA and analogies with other kinds of science. And I'm afraid that in the life sciences, we're still quite far behind in thinking about how do we create international governance schemes that actually work, that people can trust and buy into. And there's much more to be said. I don't think there are any easy answers right now.
Dr. Levy asks, how good is the serological evidence of, detection in individuals in the US preceding the awareness of the pandemic? Who were these infected individuals and what was their possible route of exposure?
That's a really good question. And I don't want to suggest I'm the expert on that story right now. The paper that I put up from Clinical Infectious Diseases from last year was a paper from the CDC. And what they did was they went to all of the regional blood donation centers, collected anonymized serum samples, and then went back and began with some screening serologic tests. Then, recognizing that some of those tests can be inaccurate, applied a whole battery of subsequent secondary confirmatory tests, and they used about 6 or 7. So I chose that paper because I think the data are a bit more sound and robust there. And again, what they found was that about 70 or 80 individuals out of many thousands had evidence of SARS-CoV-2 antibodies beginning in about mid to late December of 2019 in the United States. Unfortunately, because they were anonymous samples, we don't know whether these were people who had just traveled, people who had been near anybody who was sick, whether they were people that were sick per se, although if they were blood donors, we presume not. But you raise, of course, a much larger and somewhat contentious issue about studies done elsewhere that have suggested the same, but perhaps even further back in time in 2019. And those are studies that I think are a bit more problematic in terms of methodology and reliability. So I think I would view the likelihood that there were infections that were unrecognized both early in 2020, but also perhaps in late 2019 as certainly plausible and perhaps even somewhat likely. But what that means about the true clinical origins and where is not so clear.
Dr. Yaffe asks, if it becomes known in the scientific community, perhaps not for public dissemination, that the origin was lab-based, does this result in prominent scientists altering their view of viral genetic research? Was the Wuhan Institute a level 4 lab with adequate precautions? Thanks.
Yeah, I've written a little bit about this because I think there's a legitimate question raised. If this becomes as or more difficult than we imagine it already is, that is to gain further clarity, how important is it for us to push, given the sensitivity of all of the issues involved here? And along with that question comes the other, which is, don't we already know enough? And won't we already be looking at both natural spillovers and how to prevent them, as well as laboratory accidents and how to prevent those? And I think to some degree that's true, we will. But as someone who's followed very closely the history of laboratory safety and also risky research, and as someone who has noted the increasing power and possibility of truly consequential risky research accidents, I'm concerned that if we don't deliberately address this issue of risky research, we will find plenty of reasons not to do so. And so I think, to answer your question, yes, if greater clarity is gained, and it does look like this pandemic may have come from a laboratory or laboratory activities, then it's going to force us to look much harder. And of course, what we all fear is that there may be a backlash and there may be an overreaction. And this is where we're going to have to, I think, prepare expectations now, and where I think it's so incredibly important that we scientists and responders, I mean, all of us here on this call, start to weigh in to this discussion, because I think we have a chance now to start to shape and guide the conversation in a way that anticipates these possibilities and increases the likelihood of a productive outcome rather than just a destructive one, shutting down all research or clamping down on things that are not terribly risky, et cetera. it's an important time, and I really wish that more of my colleagues would say something.
Dr. Leibowitz asks, you have shown that risky science, quote unquote, risky science has the potential to cause pandemics. In the 6 cases you cited, has risky science been shown to prevent pandemics?
Absolutely. And so this gets back to, the whole premise behind the Global Virome Project. This was something that many of us, I think, supported, the idea of almost the moral obligation that we humans have in trying to anticipate where harm may arise and recognizing the kinds of actions that may prevent it. And then you have to ask, well, how do we do this? How do we use the tools of science to understand and mitigate risk? One of the ways that this happens is by both characterizing the viruses that arise in nature and in trying to characterize them, manipulating them. And this is what leads you right up into that little sort of gray zone, the somewhat problematic area. And I'm fully aware that it's not easy to draw lines. I mean, a lot of people have said, what about a red line? And I've actually been somebody who's proposed some discussion about red lines in scientific research, but it all comes with some peril of throwing out babies with bathwater, if you want to borrow an analogy. And I worry about that for sure.
Dr. Garcia asks, the serologic study you referenced in US blood banks is interesting. Are there similarly collected and banked samples from blood banks in China that could shed light on this?
Great question. We have to presume, yes, at least, and this is just conjecture, that many hospitals and diagnostic labs save serum samples. So if not in blood banks, and I don't presume to know what blood bank policy is in China, one would imagine that there are banked and archived serum samples. So yes, I mean, a part of the part of the WHO phase 1 investigation is said to have addressed this very possible option. And they say they sought samples and data. They were told that the Chinese had done some of that work already, and so far had found nothing prior to the dates that had been so far publicly disclosed. But the problem is we don't have the primary data. We don't have the samples or the data, the methods, the controls, and the other things that we would expect of any science project, lest alone this one, in order to develop degrees of confidence and even reproducibility.
I have this question here from Dr. Jerry Yesevich, and it's talking about, have you thought about calculating the likelihood of geographical coincidence of the outbreak in labs? He talks about the incidence, the population in Wuhan is 9 million, population in China is 1.4 billion. Independent events happening is a ratio of 9 over 1,400 or 0.006. So that happened at the same time, to multiply those two, and the likelihood of that is being very improbable. Can you comment on that?
Yeah, I'm a little bit leery about trying to do back of the envelope calculations of that sort. And here's why. There's so many factors that could play into either set of hypotheses. For example, on the natural hypothesis realm, you have to think about the movement of suspect and risky natural hosts, the wildlife trade. You then have to say, let's calculate and quantify flow of risky samples and animals. Does Wuhan, for example, happen to be, as it is, in fact, the transportation hub of central China? Might it then attract a lot more of these samples and have many more markets than other cities might? I don't know, but that's the kind of thing you'd also have to factor in. Likewise, the activities of humans. Likewise, the activities of laboratories, of which we have very little information. So, I mean, it's a good thought, and I think more of this needs to be thought through, but as yet, at least I don't know that we have the kinds of data that you'd need.
Dr. Relman, I might ask, we're almost at 9, if I could ask maybe one more question here. Are there epigenetic fingerprints that can be used to trace lab synthetically modified versus naturally divided DNA, RNA variations?
It's an interesting question. And by epigenetic, I'm assuming that someone is referring to, the sort of the true biology of epigenetics with respect to inherited but acquired markers on and features on DNA and RNA. The whole field of microbial forensics actually is an interesting one, and this kind of point has been raised. Bacteria do exhibit some features akin to epigenetics. Viruses, of course, rely upon a host cell in which they propagate, but people have looked hard at the genome of this virus and for codon usage patterns, et cetera, for any suggestive evidence or patterns of evolution, for suggestive evidence of the circumstances under which it evolved. So it's a great question. I'm not aware that there are any telltale signs, and if so, we probably aren't smart enough to recognize them because we don't have the right controls. But it's a really interesting question.
Dr. Relman, we're just hitting past 9 o'clock now. I just want to thank, first of all, the people who asked great questions. Dr. Leibowitz and Siegel, sorry for the additional questions we didn't get to. Dr. Bonilla as well. Dr. Riss, thank you for your comment as well. And thanks to all our panelists for joining as usual this week. And then finally, Dr. Relman, thank you for, again, being here today, talking about this difficult subject. And we will certainly invite you back if you have updates as we perhaps learn more. We'd love to hear more from you. But thank you so much for being with us today and for answering these questions as well. I hope everybody has a great rest of the week and holiday week as well. Thanks so much.